Hello everyone. Today we are going to discuss about the immunology and a very important topic from the immunology, that's MHC1 and MHC2, right? So we know that there are two types of MHC, MHC1 and MHC2. And we also know that MHC1 helps the cytotoxic T cell or the CD8 positive T cell to recognize its antigen, and MHC2 helps the helper T cell or the CD4 plus T cell to recognize its antigen. But how they do it? It's a bit of complicated mechanism. Let's discuss what happened actually when and how the MHC proteins are expressed from the cell. Alright, so we know that the MHC1 is expressed on all nucleated cell. Okay, all nucleated cell expresses MHC1. Alright, and we also know that our RBC lacks the nucleus. So from this discussion, you know it already that the MHC1 is not expressed from the RBCs. Alright, and what is the function of the MHC1? They present the endogenously synthesized antigens to CD8 plus cytotoxic T cell. Alright, and you might be wondering what are the endogenously synthesized antigens are. Alright, they may be some virally infected cell, and we know if some virus infects some cell and that virus produces some proteins inside the cell, maybe these proteins, or maybe there is some misfolded protein inside the cell. Okay, these things are expressed by the MHC1. Okay, but how they do it? Okay, let's just first discuss from where the MHCs are coming and how they present the antigens on their surface. Let's just draw a cell. Okay, here is a cell, and let's just see that here is the nucleus. Alright, and from the nucleus, a chromosome, maybe a chromosome that is coding for the MHC protein. Alright, and we know that the chromosome got the gene or the DNA, whatever. The DNA replicates into DNA, the DNA transcribes into RNA, and the RNA, essentially the mRNA, translated into protein. Alright, so the same thing happens for the MHC protein, and after the protein is synthesized, and I hope you know that the protein is synthesized in the ribosome. Alright, they goes inside the rough endoplasmic reticulum for some post-translational modification, and from the rough endoplasmic reticulum they go to the Golgi bodies. Alright, the Golgi bodies, and from the Golgi decides where will the protein will go. Alright, as you can guess, these MHC proteins are Major Histocompatibility Complex proteins. They are expressed on some cell surface. They should go to the cell membrane. Alright, cell membrane or the plasma membrane, and that thing is decided by the Golgi apparatus. But what happens when they are inside the rough endoplasmic reticulum? Let's just draw a big rough endoplasmic reticulum. Alright, maybe this is the rough endoplasmic reticulum. You can say it like a foot with two ends. Alright, I'm kidding. Alright, so here you can see that the MHC protein, MHC1 protein, is inside the rough endoplasmic reticulum. Alright, and they are folded almost like this, and they got some associated proteins. Maybe here's some associated protein, and here's some associated protein. And one of the important associated protein you must know, that one is the beta 2 microglobulin, beta 2 microglobulin. All right. And what happens that this protein are synthesized in the endoplasmic reticulum, but where do they get attached to their antigen? Okay, they get attached to the antigen here inside the rough endoplasmic reticulum. Before that, we must know that where from the antigens are coming. We know that the MHC1 presents the antigen that is synthesized inside our cell. Maybe a protein that is synthesized inside our cell and that protein is misfolded, alright? Or maybe some virus infects our cell and that virus produces some protein, alright? Those proteins undergo some fragmentation. The misfolded protein, they undergo fragmentation through the proteasome ubiquitin pathway, and they form some smaller proteins, and those are the endogenous antigens. Okay, and those antigens go to the rough endoplasmic reticulum, and through a transporter, they got inside the cell. All right, and what is this transporter's name? This transporter name is TAP, T-A-P, TAP. Also, you can say it transporter associated with antigen processing. Transporter associated with antigen processing. All right, then this antigen get lodged into this MHC class 1 protein, and with the antigen loaded, this MHC protein go to the Golgi apparatus, and from the Golgi they go to the cell membrane to present the antigen on their surface. All right, and then our T cell comes, the cytotoxic T cell, and then the cytotoxic T cell will induce some signals and induce the cell to undergo apoptosis. All right, that's the way our MHC class 1 protein works. Okay, now let's just take this white page and discuss how the MHC class 2 protein works. Alright, and MHC class 2 protein, they work with the CD4 positive T cell. Alright, that means they work with the helper T cells. And what they do, they present exogenously synthesized antigens like bacterial proteins to CD4 plus helper T cell. Alright, and how they do it. Okay, the same thing happens here, almost same thing. Here's a cell and here's the nucleus, and from the nucleus a gene comes out and forms a protein eventually in the rough endoplasmic reticulum that goes to the Golgi apparatus. Alright, and from the Golgi, this Golgi will decide where the protein will go. And now we are talking about the MHC class 2 protein. Alright, let's just take the Golgi, the endoplasmic reticulum, and here's the endoplasmic reticulum, and from the endoplasmic reticulum, this MHC protein inside the endoplasmic reticulum, this MHC class 2 protein is forming, and it almost look like this. Alright, it almost look like this, and this protein is also associated with some proteins, associated proteins, and here a protein binds with it, and this protein, this blue protein, is called the invariant chain. Invariant chain. Alright, and this protein, the MHC class 2 protein with the invariant chain, they will go to the Golgi apparatus, and from the Golgi apparatus they will form something called the endosome. Alright, endosome, a organelle that is membrane bound, and inside that endosome this thing will reside. Alright, the MHC class 2 protein along with its invariant chain. Alright, and now what happens, how they are loaded with the antigen. Okay, let's just guess that this is a phagocytic cell, and this phagocytic cell, and this is the phagocytic cell membrane actually, and here's some antigen that is invading the the phagocytic cell. And from the shape of the antigen, I hope you can guess that this rod-shaped organism, maybe some Staph aureus, sorry, maybe some Escherichia coli that is invading this, and whenever it get inside or the phagocytic cell, the phagocytes it or it engulfs it, it will go inside the cell. It will go inside the cell and it will form something called phagosome, and it will fuse with something called the lysosome. It will fuse with something called the lysosome. And we know that the lysosome is some acid molecules are inside the lysosome that acidifies some other particle or other proteins. So these two will fuse, and along with this endosome, this phagosome and this lysosome will fuse together and form a bigger endosome, all right. And here, as you can guess, the lysosome, this secretes some acidified molecule. This thing will be acidified, acidic environment will be there, and due to that acidic environment, that MHC protein, MHC class 2 protein, they will get rid of their invariant chain. Their invariant chain will be detached from them, alright. And this thing, this bacteria or foreign protein, they will also be cleaved inside the acidified environment, and the antigen will be bound here, alright. And the whole thing will go to the cell membrane, and they will enter the cell membrane or transmembrane, and they will present this MHC class 2 will present outside the cell membrane, and they will present the antigen outside, alright, like this. And that's how they will present the antigen to the T helper cell. I hope you enjoyed the video, and if you want to get tutored, I'm a tutor. I tutor over the Skype, and I use this interface for tutoring. If you want to get tutor, please call at my number, and or you can also email me.